Welcome students to today's module of e-learning. I am Dr. Maria A. D'Souza, Assistant Professor, St. Xavier's College, will present this module to the first year students of Botany, Semester 1, BOC 101, course title Biodiversity 1, Microbes, Algae, Fungi and Bryophytes. The title of the module, Unit 3, Fungi, module name, Agaricus, Reproduction. The outline of the module, Reproduction in Agaricus and the life cycle of Agaricus. The learning outcomes. Once the student completes this module, the student will describe the method of reproduction in Agaricus, explain the terms plasmogamy, karyogamy, meiosis, and finally illustrate the life cycle of Agaricus. The reproduction in Agaricus is 3 types: vegetative, asexual, sexual. In the sexual reproduction, the Agaricus doesn't form definite sex organs. Vegetative reproduction. The Agaricus species vegetatively propagate by means of perinating mycelium, and it happens in most of the edible species of Agaricus, where small pieces of dikaryotic mycelium are used as inoculum. The pieces are grown in soil-rich organic manure to obtain the basidiocarps. Asexual reproduction is very rare. It reproduces by means of chlamydospores, oidea, sclerotia. Chlamydospores are terminal intercalary in position on the secondary mycelium, and it is responsible for forming the dikaryotic mycelium. Oidea are mainly involved in dikaryotization rather than developing into new mycelia. And the third one, we have sclerotia, though this method is not very common in Agaricus. Sexual reproduction in Agaricus takes place by a method called as somatogamy. Most of the species in Agaricus are heterothallic, except for Agaricus bisporus, which is homothallic. Somatogamy is achieved through 3 steps: plasmogamy, karyogamy, and meiosis. Plasmogamy is the fusion of 2 compatible hyphae. That means 2 primary mycelium, a plus strain and a negative strain, come and fuse with each other, and the nuclei do not fuse, students, and it results into a binucleate cell called as a dikaryon. This dikaryon undergoes successive divisions and forms the dikaryotic mycelium by means of clamp connections. This dikaryotic mycelium is perennial. Perennial means very long-lived, subterranean, and produces basidiocarps under favorable condition. So students, this is the picture to show you the primary mycelium. This is the primary mycelium fused together, the dikaryon. Successive divisions through clamp collections will form the secondary mycelium, which will form the tertiary mycelium, which will give rise to the basidiocarps. So plasmogamy, it is followed by a karyogamy, wherein both nuclei of dikaryon fuse to form a diploid nucleus, and it takes place in young basidium. Immediately after karyogamy, meiosis is done. And by the process of meiosis, the basidiospores are formed, which are haploid in nature. Now let us see the development of the basidiocarp. The development of the basidiocarp takes place by subterranean mycelium called as the rhizomorph. It arises as thin, tiny, white apical swellings consisting of dense knot of hyphae of dikaryotic mycelium. Now, this mycelium, the rhizomorphs, takes in sufficient nutrients, and once it has taken in sufficient nutrients, these tiny white knots enlarge. They come up, break through the soil, and they are called as the basidiocarp primordia. They enlarge further, they become round or ovoid structures, and this stage is called as the button stage. If one takes the longitudinal section of a button stage, it shows differentiation into two regions, the basal part or the stalk region, which will form the stipe, and the upper expanded portion, which at maturity will form the pileus region. A ring-like cavity called as a gill chamber also develops at the junction of the stalk and the pileus region, and it is called as the prelamellar chamber, which will give rise to the future gills. Now, when the mushroom is small, it's in the button stage, it is covered with a membrane which is called as a velum. But as the button mushroom expands, it grows, it becomes bigger in size, the growth proceeds more rapidly at the upper region, forming an umbrella-like portion which is called as a pileus, whereas the growth doesn't take place that much faster pace in the stipe region. Now the velum ruptures itself and it exposes the hymenial layer or the gills or the lamellae. The gills, when they are exposed, they are white in color, but later on as they mature, they change to pink, then to dark brown. According to Atkins in the year 1906, he called the development of the basidiocarp as hemiangiocarpic. Students, hemiangiocarpic means that the hymenial layer is initially enclosed by a membrane, but on maturity, it becomes exposed. Now, at the same time, the veil which is there, which is covering the velum which is there, which is covering the button mushroom, as the mushroom increases in size, it breaks open and the remnants of that velum remains attached to the stipe, forming the annulus. So this is how, students, this is the button mushroom stage. It starts forming the prelamellar chamber. Then it increases in size. The growth takes more faster in the apical portion, forming an expanded portion, which is the pileus. On the lower surface, you have the gills. The velum remains attached, hanging like a collar, and it forms the annulus, and this is the stipe. Now, if you see the development of the basidium and the basidiospores, they are formed on the undersurface of the gills. Where the gills, if you take a section, you will see three layers, students: strama, subhymenium, hymenium. The hymenium will produce the basidia, and the basidia are the spore-producing bodies. And if you see, the young basidium is a dikaryon. It is arising from the subhymenial layer, and it is binucleate cells. The nuclei, the two nuclei inside the basidium, fuse and form a synkaryon. Karyogamy is achieved, and you have a diplophase. This phase wherein karyogamy is achieved, a synkaryon is formed, is ephemeral, students. That means it is very short-lived. As soon as it is formed, karyogamy is followed by meiosis, wherein the nuclei divides into having 4 haploid nuclei. Okay, and it forms the basidiospores. Okay, now simultaneously you will have 4 peg-like structures arising from the distal end of the basidium, and it forms a small basidiospores as budding out from that peg-like structure. That peg-like structure is called as a sterigmata. A large vacuole also develops in the basidium, pushes the cytoplasm towards the peg-like portion, towards the small basidiospores, and one nucleus each migrate along with a little cytoplasm in the basidiospores. And we get 4 basidiospores in each basidium. 2 of a plus strain and 2 of a negative strain. So in young condition, the young basidiospore is unpigmented, but later on, it can change its color as it matures to brown or black pigment. So students, this is how the development takes place. The dikaryon, fusion, the karyogamy, followed by meiosis. Meiosis cytoplasm, the vacuole increases in size, pushes each nuclei inside, small peg-like structure, sterigmata, and this is how you have the formation of the basidiospores, wherein 2 basidiospores are one type and other 2 are of another strain. So this is how it looks, students. You have the trama region of the gill, you have the subhymenial region, and the fertile region, which is the hymenium, which will bear the basidium, the paraphysis, sterile ones, peg-like structures, sterigmata, and the basidiospores. So once the spores are matured, they have a mechanism by which they will shoot them out. And that mechanism, discharge mechanism, is called as water drop mechanism, water bubble method, or Buller's drop method, wherein in this method a small water droplet appears and it creates a pressure there and pushes out the basidiospore. The basidiospore is uninucleate, has a wall of chitin and chitosan. They are very light in weight and they are shot horizontally and in rapid succession, from where they will fall vertically downwards. Once the basidiospore falls on a substratum, it will start germinating. And if the basidiospore is of a plus strain, it will give rise to a mycelium, which is of a plus strain. If it's of a negative strain, it will give rise to a negative strain primary mycelium. In any case, it is monokaryotic. That means it has a single nucleus. Now, these mycelia, plus strain and negative strain primary mycelium, will fuse and form again the secondary mycelium. And by means of somatogamous copulation, they will form the reproductive structures, the basidiospores, and finally the basidiocarps. So if you see the life cycle together, students, the life cycle in Agaricus shows 3 phases: vegetative, asexual, sexual. Vegetative is by means of perinating mycelium. Asexual, which is very rare, students, by means of chlamydospores, oidea, sclerotia. But the sexual reproduction produces sexual spores called as basidiospores. And it starts from the basidiocarp. On the lower surface of the basidiocarp, you will have lamellae or gills, which will produce the basidia on the hymenial layer, the fertile layer. The basidia will undergo meiosis, karyogamy takes place, undergoes meiosis, 4 basidiospores, 2 of plus strain, 2 of negative, which will start germinating from primary mycelium, pus strain, negative strain. Fusion of this 2 mycelium, dikaryotization takes place, clamp connections will be formed, and then the secondary mycelium is very much long-lived, which will give rise to the rhizome of the tertiary mycelium, and then again will form the basidiocarp. Basidiocarp will give rise to the basidiospores. And this is how, students, the life cycle is completed. If you see, students, the diplo phase is very, very short-lived, followed by the haplo phase, but the dikaryo phase in Agaricus is very, very long-lived. So in this way, Agaricus completes its life cycle. So to summarize, students, today we have learned the different modes of reproduction in Agaricus, which includes vegetative, asexual, which is rare, but if it happens, it will happen through by means of chlamydospores, oidea, or sclerotia. And finally, the sexual spores, the basidiospores, by means of sexual reproduction on the gills which are there, which are on the undersurface of the basidiocarp, by means of plasmogamy, karyogamy, and meiosis. The process is called as somatogamy. So in this way, the Agaricus, the mushroom, will complete its life cycle. So I hope students, you have understood it. These are the references, students. The offline references and the online. And thank you, students, for a patient hearing.